So this is kind of where we left off in class, where we were talking about what proteomics is. And proteomics is just a system of study where you can look at all these proteins. So it's a full set of proteins. And a lot of times you can kind of mark which ones those are. When I was in grad school, I was looking at protein changes associated with human heart disease in the mitochondria. So I'd extract out the mitochondria, and then I would kind of break it down, digest it down, and run these things on pro these gels, kind of not using the same idea that we did in class and lab, but they look slightly different. And then you can compare, and what's so cool about this is, say for instance, you have some kind of disorder. You can take the protein picture of all the different proteins of what they look like in a normal person and then compare them to someone who has a disease. See if maybe proteins are there that weren't there in a normal person or are absent now. And that can kind of give you an idea to start investigating those changes. And so we have to understand what kind of proteins are produced in there to understand the function of cells and organisms. Because if you remember, proteins are essentially components that make things up, or they're enzymes. So they're the scaffolding of a lot of things, or they're the doers and shakers in the cell. So in either case, if either one of those are broken, or say for instance you have something doing something in the cell that's not normally there, that can cause problems, that helps us understand it. And when we look at the changes of behavior for a whole system, that's what's known as systems biology. And that's essentially how biology is going. For so long, we've been looking at just really small parts of organisms or cells and been very specific on what's happening with this one thing. But now we're trying to look, since we've done so much investigation like that, we're trying to look a little bit bigger and say, okay, what's happening in the whole cell? It's a difference between looking, okay, there's a dysfunction, there's something happening in my toilet, versus if your whole house is messed up. Let's look at if something's flooding. Okay, instead of just looking at the toilet, let's look at the effects it's causing in the whole house. Maybe it's on the second floor and it's dripping down to the first floor. So it's kind of saying, looking at the whole house and how it functions to get an idea of what houses are like, versus looking at just the toilet in the house and how it functions, and then trying to make assessments based on just that toilet. You see how that can be kind of a little bit harder to do? So systems biology is more like looking at the whole house and how it functions. And basically, systems biology took this approach on several different medical applications. One specifically is looking at cancer. And so the Cancer Genome Atlas project was completed in 2010, and we talked about, and it basically attempted to identify all the common mutations in 3 types of cancer. And so mutations are just changes in DNA, and they can compare by gene sequence and expression and the cancer models versus normal models. So the normal models may show that these particular DNA sequences have not been changed, and then they can look at it and identify those changes that they've seen in cancer cells, specifically looking at DNA. And so this was so fruitful, and one of the examples is the cancer associated with breast cancer and being able to see mutations in genes there, that essentially they've been able to extend these to 10 other types of common cancers. And so what they've used to make this very automated and quick is silicone and glass chips, and they produce that hold a microarray of most known human genes. And so this is what it looks like. It's a gene chip, and a microarray is just what you think an array is, a bunch of different things, and micro is small, right? And so if you look at here, this is where all the information is going to be processed. And right there is basically a tool that has a bunch of different gene sequences that you can basically do this analysis with and determine what is normal and what is not normal in that little spot. Pretty cool stuff. So when we're looking at microarrays, a lot of times we're looking at large genomes. And genomes are all the different genes in an organism. So by about August 2012, there were 3,700 genomes that have been completely sequenced. And most of those you can find in the NCBI site that you use for your homework if you did that. That includes about 3,300 bacteria, 160 archaeal, which are the ancient bacteria, and about 183 eukaryotic genomes. As you notice, there's a lot less eukaryotic genomes than bacteria, and a lot of that is the overall number, but also the bacterial archaeal genomes are so much smaller than most eukaryotic genomes. Sequencing of about over 7,500 genomes and about 340 metagenomes are in progress right now, and that number's actually increased since that the number completed and the number that we're looking at. And so if you look, the genomes for bacteria and archaea range between 1 and 6 million base pairs, where eukaryotes are much longer, which kind of helps you understand why there's not as many that have been sequenced. Most plant and animal genomes are greater than 100 million base pairs. Humans have 3,000 of these million base pairs, right? So you can see why it took so long to actually sequence our genome. Within each domain, there's no systematic relationship between genome size and phenotype. And what that means is just because that there may be a lot of base pairs doesn't mean that that means there's going to be a lot more proteins or the phenotypic differences that you see is going to be greater. And so as we kind of looked in class, just a little bit more review, you can see here the number of genes and size. So here, this guy right here, you can see is a common E. coli. So the genome size is 4.6 million base pairs. The number of genes is 4,400, so 4,400. And the genes per million base pairs is about 950, right? And then you can see over here that our size is 3,000 million base pairs. And the number of genes we have are greater than 2,100. So we do have more genes, but do you see genes per base pair is much smaller here, right? So we have a lot of basically junk DNA, right? That we right now have no idea what they're coding for. And you can see here we have around 2,100. And then if you look at here, this is just a mustard plant, right? Arabidopsis here, and it has 27,400 number of genes, right? And it's 120. So here we have 228 genes per million base pairs. So they actually, more of their genes or their DNA actually codes for proteins, where there's a lot of us still unknown. And so this kind of gives you an idea on average that free-living bacteria and archaea bacteria have about 1,500 to 7,500 genes, where basically basic fungi have about 5,000. And most multicellular eukaryotic cells have up to about 4,000 genes. So the bigger the organism, most times the more genes, but as you can see earlier, that's not always the case because mustard plants are very much smaller than us. And a lot of that has to do with just still not understanding the human genome yet because it's so complicated. Now, when we want to go and make changes, like we've talked about, for making human insulin using bacteria or changing different plants, there is this thing called recombination, and it's tools using recombinant DNA. So you can mix DNA up, right? Like I was talking about in grad school, where you can take a gene that's bioluminescent in jellyfish and put it into a mouse. Heck, we could even put it into, like they said, dogs and cats, and work on putting them into us if we wanted to. And it takes a few things. It takes what's called a plasmid. If you remember, that's circular bacterial DNA. Restriction enzymes, these break DNA into smaller sequences. Gel electrophoresis, which we used in lab, right? And that is a separation technique. And PCR, which is called polymerase chain reaction. You should notice this word right here. In PCR, basically takes little amount of DNA and makes lots of it. So this is what's helped if you like to watch CSI, or if you like to watch SVU and they're doing DNA tests. This is what essentially allowed us to start being able to do that. Because if you get blood, there's such a small amount of DNA there that there wouldn't be enough a lot of times in cases to run it and be able to identify DNA. But with polymerase chain reaction technique, you can. So as I was talking about, plasmids are pieces of DNA, and what's so cool about them is that you can customize them. So if gene cloning is one application of DNA technology, and basically gene cloning is being able to take a piece of gene and make more of it, right? And so we have gotten really good at making methods for studying and manipulating gene material using plasmids and gene cloning. So how this works is a lot, basically the process of transformation that we talked about, right? So here's our gene of interest, right? This could be us. This could be the human insulin gene here, right? And so they take out, and this is the DNA, and they cut out the gene right here. And this is a plasmid, that circular piece of DNA and bacteria that they take out. And basically they mix the two together. And so you see this is the part of the regular plasma, and this is over here, this is your what's called GOI, or gene of interest. And then what they do is this gene of interest and this plasmid together makes what's called recombinant DNA. And they put that inside a bacteria. And then that bacteria will divide, right, using binary fission. And then basically, once it's out here, it'll start making basically proteins. So if you put this into a plant, right, and this gene made the crop somehow resistant to pests or insects, right? It's gonna basically, this gene will help keep them alive. This is how Monsanto was able to create plants that were Roundup resistant. They basically make a protein that makes Roundup not toxic to them. You can also do this. Genes have been used to alter bacteria to help clean up toxic waste. So they add a gene in there that'll help to basically eat up or break down whatever toxic waste is there. Now they've also made it copies to help with human diseases, right? They can use proteins to put in there that basically dissolve blood clots in heart attack therapy. You can talk about proteins used to make snow form at higher temperatures and change. So there's all of these different ways that you can clone cells, which is make copies of them, by having a gene of interest in them. And this is a perfect example. This is how they were able also to make insulin. So when they do this, when they have that gene of interest, they take it out. But the way that, remember, it showed you initially that piece of DNA, there was a lot of it, right? And we want to take a small part of it. So what they do is, if this is the gene of interest, right, they'll get something called a restriction enzyme. So what the restriction enzyme will do, it will cut the DNA at certain parts. So here you're seeing, and some of the names are like EcoR1. You might see there's a bunch of BAM-HI. If you ever see that written anywhere, and you don't need to know those names, right? It's just those are restriction enzymes, and they cut at certain parts. So if you see here, do you see how it's cutting between the GA here and the GA here? So you can see, essentially, that's basically where they're going. That's a restriction enzyme that's showing where it cuts, right? And it makes these things that are called sticky ends. So there's two types. There's either blunt ends, where the DNA is cut straight through, or there are these sticky ends, if you see right here, where it hangs over. This is blunt. This is sticky. Blunt, just like it says, it cuts it, makes a blunt edge. Sticky is named this way because this is going to make it easier to attach to a new piece of DNA. So if this is your, basically, this is your plasmid here, right? This is your gene of interest. You will cut both with the same restriction enzyme. Restriction enzyme, RE, right? So that when you mix them together, the overhangs will be the same. And so it can come in, and then you just use the ligase, just like before, right, to make sure that they bond together. So restriction enzymes are used to basically cut, remove pieces of DNA, and then put the new piece of DNA back in. Now, polymerase chain reaction is important because this is what's going to, also known as PCR, is a technique that is going to amplify any piece of DNA. And you don't need to use cells for it. Basically, you have a very special DNA polymerase. And essentially, it's a polymerase they found in bacteria that were in the hot springs. And essentially with this, it was able, you mix your piece of DNA with a supply of nucleotides, right, with the DNA polymerase, and basically there's a short piece of single-stranded DNA that's known as a primer. You make this. Primers, remember, help start. And then basically after this, you can make billions of copies of DNA within a few hours. It's very, 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 very fast, right? And so this is what basically revolutionized molecular biology and forensics biology. So basically in PCR, there's 3 steps. There's a heating, a cooling, and replication. And that's going to bring around a chain of reaction that produces exponential growth, right? So this idea is that you basically start and you kind of grow exponentially, right? So basically what it starts with is the reaction is first, that reaction mixture is first heated, and that's going to help denature the DNA strands from each other. Then the mixture is cooled. So first heat, then cooled. And that allows hydrogen bonding of the short single-stranded DNA primers that are complementary to the sequence on the opposite side of each target that you're trying to start, right? So basically what you're doing is you're starting out. Give me a second. Move this really. So you're starting out with 2 pieces of DNA, right? Then you heat, which is going to make them separate. And then what you're going to do is cool. So basically, your small primers are going to attach. And then you're going to heat together again, and then what's going to happen is that the DNA polymerase is going to attach, and then you're going to be able to make the sequences. We go DNA polymerase attaches, right? And so that's essentially what's happening here. Oops, sorry guys. And so, if the standard DNA polymerase was used that wasn't used to the heat, the protein would denature during the heating step. So that's why the special DNA polymerase, the discovery of that, was very, very pivotal for this process. So let me see. Let me get out of here. So this is basically how the process works in a nicer picture, right? So this is a genomic DNA, and this is the target sequence that you cut up, and you're ready for it to, basically for you to use, right? And so first you heat it. So this is heating, and you can see it separates, right? This is cooling, so you can see the primers get on there. And then this is heating, and this is where the new strand comes, because this is where the DNA polymerase starts working. And then you do the second cycle again, right? So then you are going to cool, and you have the new heat thing, right? And then you heat again, and then you cool and heat. And so you have this cycle. And after, if you have one at the first, at the end, because you split it in half, you're going to have 4, right? And then 4, you're going to double that. And so you have 8. And then 8, you're going to have 16, 16, 32. And it's going to keep going up exponentially. So you get a lot. And this is so cool because they've been able to do a bunch of different things. When they found 40,000-year-old frozen woolly mammoth cells, they were able to take that ancient DNA and amplify it by PCR to have enough so that they could do all kinds of really cool tests on it. So that's pretty awesome. And so what they can do once they have so normally what they do is, we've talked about 2 of these things, right? We talked about plasmid being in DNA. We've talked about our genes of interest. We've talked about the sticky ends with the restriction enzymes. We've talked about PCR. And then we've talked about just basic transformation. So how all this works when you put it together, guys, right? So this is chromosomal DNA. This is the plasmid DNA, right? And so basically you're going to isolate this plasmid DNA, right? Which means you're going to take it out of this bacteria. And then you're going to isolate the human DNA. And basically what you can do is a couple things. You can get your restriction enzymes, right? And you're going to cut these, right, with restriction enzyme, and cut this. You can also do PCR on this so that you have enough of your gene of interest for your human gene. And then what you're going to do is you cut both with the same restriction enzymes. Can you see these guys have the sticky ends both for the human DNA and for the plasmid DNA? So then they can recombine, and they're called recombinant DNA. And if you can see, there's a couple different ways that they can recombine, depending on how they get cut. Here, they recombine with the human DNA fragment or the gene of interest that you wanted, right? Here, wait, let me see. Actually, the black. So the black here is the gene of interest. Let me go back here. Sorry. I thought it was the pink. And so it can recombine a couple different ways. So this is all of the different, let me clean this out so we can start over. These are all the different human genes. So the black one is the gene of interest, and these other ones are just other fragments that were cut as well, right? So they'll have sticky ends as well. So when you mix them all in, you'll have this one right here, which is the gene of interest, and then you'll have these other 2. And both these other 2 just have other genes from human that you don't want. So how do you end up determining which bacteria actually gets this guy, because this is the one you want, right? Well, if you look on here, they actually have little selection markers on here, and this is AMP-R, which basically says this plasmid has a gene on it that has a protein that will make this guy ampicillin resistant. Ampicillin is an antibiotic. So essentially what happens is only the cells that have the plasmid will grow. So if you have a cell that just has bacterial DNA but did not take up the plasmid, they won't grow when you plant them on here. Only these guys will grow. And then essentially what you'll do is, how can you tell if it's this particular one here? Well, in that, pretty much that guy, you will make it so there's another selection. And in here, the ones that are actually incorporating this gene will be the white colonies, and the ones that don't will be the blue. So there's a color aspect on here that basically selects this. And it's because the gene that you put in there will actually disrupt the gene that normally makes the blue color, right? So the big part about this is knowing that plasmids have a way to select, right? And what I mean by selecting is they have a way to, first, if the plasmid's on here, it will grow because it's resistance to ampicillin. All the ones that don't take in the plasmid won't grow. And then if you want your gene of interest, it'll disrupt some of the genes that are already on the plasmid, so it'll make it so that they don't produce that blue color. So they can select for both the gene that you want to look at and the plasmid itself. So that's can be a little complicated there, but that's essentially how it works. And you need PCR, and you need recombinant DNA and restriction enzymes to do that. And so in 1982, that's when they started human insulin by bacteria. And that was actually the very first recombinant drug approved by the Food and Drug Administration. And it helped to make the treatment of diabetes reasonable, financially reasonable for the general population. And so this is just, I'm looking at how restriction fragments, or these small fragments, can reflect DNA sequencing. So these things called restriction fragment length polymorphisms. Polymorphism, think if you morph into something different, right? It's a change. So poly meaning a lot. So these guys can actually reflect differences in sequences of DNA samples for humans. And so if you look, you can compare these guys, these RFLPs, at a crime scene to a suspect and see if they're the same, right? And so they'll use kind of these restriction enzymes to cut, and then they'll look at the fragments to see if they're similar. This is how they also can look. These are the small fragments of DNA that are used to see if people are on the crime scene are the same, if you're doing paternity tests. Microarrays can be used, tests similar that we were talking about earlier with the cancer chip, is can reveal basically patterns of gene expression in different kinds of cells. So not only could it be used for cancer, but it can test many genes at one time. And so how this works, just to explain earlier kind of what was going on earlier, that you can take DNA, reverse transcribe it like we were talking earlier, since we know about those enzymes now, to make RNA. Or not RNA here, but complementary DNA, cDNA is what it's called. And then basically you take that cDNA and you put it and mix it with the DNA of an expressed gene. And so what happens is if the cDNA attaches to the blue or the original expressed gene, then it lights up and turns, in this case, yellow or green up here. And if there's no complementary DNA that you're looking at, then this gene is not expressed, so it doesn't glow, and so here is blue. And most of the time you can see these in red and green. So this is actually a DNA microarray, right? And basically it'll have here is an example of 6,400 genes. This is the actual size, right? And the green will tell you, yes, it's expressed, and the red will tell you it's not. And it's all because you just take DNA, you make the complementary to the ones it's actually making, the genes it has, and then when they bind together, it basically says it's a positive that it's there. If it's not there, then it's a negative that it's not. And so you can test large amounts of genes with this. Now, another way you can test genes using those polymorphisms is like what we did earlier, the gel electrophoresis. So you can get a mixture of DNA in here, right? You put them in those little wells, and you have a power source. And DNA is negative in nature. And that's because of all the phosphates. Remember, you have the phosphate backbone? It's phosphates and oxygen on there. So those make basically the backbone negative. So when you put them in here, the DNA is negative, so this negative charge is going to push it, or the positive will pull it to its side. And so all the DNA is going to move this way. Now, what makes the DNA separate out more? It's not their charge. It's that these ones that go out faster are smaller, right? Fragments of DNA. And these are longer fragments, right? And so then you can look at here, and then what happens is you can compare them. So if you're looking at this is one sample, this is the second sample, this is the third sample, right? Then you can look and you can go, well, this one matches, this one doesn't, right? This one matches. This one matches for all three. This one matches here. This one matches all the way across. This and this match, right? These two match. These match here, right? And so you can see that these samples essentially have some commonalities between them. So we do this by mixing them with restriction enzymes that break them up into smaller pieces, and then they can walk through. So this guy that's at the top, this is going to be a larger piece of DNA, and this is going to be a smaller or shorter piece of DNA. And then you can look at them, which is what we did in lab, right? And so these restriction fragment length polymorphisms that we can talk about can detect differences in DNA sequences, and they're basically revealed on bands. Now, when we're looking at harmful alleles or anything that could be disease-wise, a lot of times what you can do is use radioactive probes, and those can reveal your gene of interest. So essentially how this works is here you're taking those three sequences again, you're running them on a gel, right? And then what you're doing is you're doing a blot procedure. So you're taking the gel, you're laying it on top of this filter paper, and you're moving the DNA, from gel to paper, right? And then what you do is once it's on the paper, you can label it with radioactive probes. That way, then you can essentially take a picture of it using something like video or film, right? And so, let me get out of here really quick. So this is something called a blot procedure, and there's different types. You basically have northern blot, that can be used for this way to detect basically RNA. You have southern blot, to detect DNA. And then you have western blot. What do you think that can detect? If you said protein, you would be right. So these are different things that you can look at to either uncover DNA differences, RNA differences, or protein differences. Now this, if you're doing DNA technology, it can be used in a court of law. And initially back in the 80s and even early 90s, there were a lot of times where this was not admissible in court. Many violent crimes in the past lacked enough evidence to be able to solve them. But if there's biological fluid left in the crime scene, later it could be isolated for some of them and figure out who did it. Now, as you realized in the past, HPD, or the Houston Crime Line, has had some problems with people either contaminating it or basically making false positives. So it's something that if you go into it or you think it's interesting, you definitely have to be very good at your job, make sure things do not get contaminated, and very ethical. DNA fingerprinting is a set of laboratory procedures that determine with near certainty whether two samples of DNA are from the same individual. So it's very, very good process to use, and it's been a powerful tool for crime scene investigators. You just have to be careful. Like I said, if you contaminate that DNA at all, it can give you false positives or false negatives. Or if you, this is the time where you have to really trust the person that you're doing it with, because they can give false positives if they feel like just somebody is guilty. It's been seen in the past for that to happen, that they will doctor it to make them guilty. So that's why you have to be very, very careful on the data that you use and make sure that everybody's doing what they're supposed to. So with DNA fingerprinting, what happens first is we cut it with restriction enzymes. And then we're going to separate the fragments, and we do that again by electrophoresis, that process that you did in class. And so we place it in a gel, we move it with an electrical current, and that's going to move the fragments through the gel and separate them according to their size. And the DNA will form bands, and those bands will correspond to bases. And for these polymorphisms that we have, no two people have the exact same sequence of bases in the gel. So that makes it very unique for individuals. And so this is the idea that I showed in class, that you can have this defendant and here's the victim's blood. And these are the DNA bands. And this is the blood that was found on the defendant's clothes. Here is the jeans and here are the shirts. So you go through and you look and you can see that some of these are here. And well, that has the same band for him. But if you look, this band, and this band are all the same. If you look on the jeans, it's the same. But if you look over here, it is. Here is the same. But again, you can see over here on that one. And then if you look, nothing, right? Nada. Here, nada, but these both are there. And so this kind of tells you here that this right here, this shirt, the blood on it is from the victim, and that would help them to identify. And if you see, it's this color. So essentially, this is one of those processes for which they use probably at blotting to do this. Now, what else is cool with recombinant DNA and how they can do? In 1997, and I remember being really young when this came out, young as in just finishing high school, and the birth of Dolly was announced. And Dolly was the first animal that they've cloned, and they've been able to clone other things. So basically, Dolly was a lamb clone from an adult sheep by nuclear transplantation from what's known as a differentiated cell. The thing is, Dolly died pretty young. She died in 2003, and her arthritis had led to speculation that her cells were not healthy as those of a normal sheep and probably reflect incomplete programming of the original transplanted nucleus. So this they still have not been able to really do well, but you can get things like they've talked about cloning pets so that you can get your original pet back, right? So this is the last thing we're going to talk about, and it's pretty interesting. So what we've done is you have basically, here's the sheep that we're going to clone into, right? So we take an egg, we take out the nucleus. So at this point, we have no DNA in this cell, right? And then what we do is we take cells from the mammary cells of this guy right here, and we put them on a plate and started growing them, right? And then in the middle of the cell cycle, we're going to stop it. And so that's going to cause them to de-differentiate. And so differentiated cells are basically cells that have decided what they want to be. They're like, okay, I am a skin cell. That's what I'm going to be. So during regular functions, they're acting as a skin cell. But they stop acting like a skin cell really quick during the time that they're dividing. Because it doesn't matter. They're dividing, right? So when they arrest it and they stop that during that time, they basically put it back to just being a normal cell that doesn't have a function. And they take the nucleus and they're going to fuse that cell with the cell that doesn't have a nucleus. And then what they're doing basically is putting the entire genome or that nucleus from this mammary cell into this egg cell from the ovary that doesn't have nucleus. And then this is grown in culture, which means it's grown like that on a plate. And then as it becomes an early embryo, here, they're going to implant it in the uterus of a third sheep. And this is basically the surrogate mom. And then basically what happened with the sheep, it basically allowed it to grow inside, and then it was born, and Dolly the lamb basically was genetically identical to the original mammary cell donor that was here. Now the problem is it's not perfect, and there's still some flaws in it because Dolly was somewhat sick from the process, but they're still working on to make it better. And so that's essentially everything about DNA, and I hope you enjoyed it. And that's the last class, and everything's finished. So I hope you have a great afternoon, guys, and I'll talk to you soon.